Hi, I'm Rebecca. I'm a third year PhD student in the Quantitative and Applied Ecology Research Group at the University of Melbourne. Today I'll be presenting my chapter investigating the use of spatial count density estimates of cats and foxes in a management scenario. I'd like to begin by acknowledging the traditional owners of the land on which this research was conducted and where I am today, the people of the Antakarudinye Martu Yukuntra Nation and the Wurundjeri and Bunwurrung peoples of the Kulin Nations. I pay my respects to their elders past and present and to any other communities in our virtual room. That gorgeous sunrise was at Bush Heritage, Bonbon Station Reserve in the arid rangelands of South Australia. Whilst it doesn't look too far from the coast here, unfortunately, internet and mail services are unreliable at the best of times, let alone at present. Consequently, data transfer and model fitting has severely slowed, so I'll only be able to present preliminary results from a subset of the data today. To benefit extant fauna, an intensive predator management plan is underway. My task is to estimate cat and fox density inside and outside the predator management area. Ultimately, we want to understand the level of management required to maintain suppression of fox and cat populations and to facilitate potential reintroductions of species such as the booty that you can see pictured here in the bottom left. Working on this in a sparsely populated, lower productivity region presents several challenges. We want to use methods that accommodate these challenges, but also provide a more complete picture than, say, using activity indices alone. So to do this, we have set up two camera grids of 48 cameras, spaced 1 kilometre between each camera. Although the landscape looks quite homogeneous at this scale, there are quite a few different habitat types, and we've put the two grids in similar habitat. We space them as far apart as possible so that frequent migration between the two is unlikely. The cameras are operated continuously. Here is a timeline showing the operation of the grids. The northern grid was established prior to my PhD, and we started the second grid 2 years ago. The solid line indicates when no predator control is occurring, and the dotted shows when predator controls begin. Initially, we started with ground baiting for foxes, and then we increasingly trapped and shot both cats and foxes. Southern grid has no baiting at any time. So this totaled over 80,000 trap nights, or over a trap double century. I have to acknowledge the massive effort in camera data sorting and management by Kate Taylor. Due to environmental conditions and distances, our cameras are also unbaited. Consequently, we are haunted at night by lots of ginger ghost cats. So identifying cats is difficult, and the risk of misidentification and double counting is too high. So we don't have any individual cats identified. As for foxes, they might sometimes stop and smile at the camera, but they pretty much all look identical. So I'm exploring the use of unmarked spatial count models to estimate the density of both cats and foxes. Here is an aerial view of the southern camera grid area. The dots represent the location of 12 foxes during one session. For us, that's a month. So the foxes are running around, hopefully going in front of our cameras, but some of them won't be detected either. These locations are latent. We don't know how many there are or where they are. We're going to estimate this from our camera traps. So how do we do that? Let's step back from the population for a moment and think about a single fox in our environment. It's surrounded by its area of activity, which we assume to be circular. The center of its range will be used more frequently than the edges, so we would expect to detect it more frequently if a camera is located close to its center of activity. This forms our detection function, with lambda naught being the expected number of detections if we happen to place a relay camera right next to a fox's activity center. So let's imagine that we're able to catch this fox and mark it green. If we identified our fox 6 times in the yellow circle, 3 times in the green, and once at each of the other cameras, we could estimate its activity center to be about where the small blue dot is. If we can do this for most or all of the individuals we detect, we can see how we build up a picture of the whole population and the density. So that's great, but foxes are really hard to catch, trust me. What our data actually looks like is a count of foxes detected at least 1 hour apart at each camera. But how do we turn these different sized dots into an estimate of density? By placing our cameras closely together, we induce spatial correlation in our counts. One fox can be detected at multiple cameras. We use this correlation alongside our understanding of fox movement to estimate whether detections are of one or multiple individuals. We can use covariates as well, for example, the distance to track that a camera is placed. And this can change the shape of our detection function, like this, for example. Because we don't have any individual identification, it's more difficult to estimate the detection parameters. But what we do have is information on their range of movement, which is related to sigma. From published home range data, I estimated a gamma distribution for sigma, excluding outliers. Here you can see the estimates of sigma from published home ranges in similar environments to Bonbon in the black dots. In a previous chapter, I compared spatial count estimates to published spatial mark resite estimates of foxes with very promising results. So I've applied these models here. I shared detection parameters across time and across the two grids, so we are able to have density estimates even when no predators are detected as populations shrink with control. I have additionally included a categorical effect of predator control intensity on the population size. I ran into many memory issues with JAGs, so my models have been fit in Nimble. Thanks for developing this, Perry. So I'll just reiterate that these results are very preliminary and I only have autumn and spring subsets of the data at the moment. Models with the full data set to November last year are currently running, so we have some no tweet signs coming up. I'd love to hear your thoughts about how I've approached this problem and whether you have any other ideas for analysis of this data set. So I'll start with the raw number of detections in each grid. The grayed out area indicates the period of intense predator control in the northern grid, and please note the dates differ between the two grids. Here we have the raw counts of foxes in black and cats in white. We can see that there's quite a bit of noise in the raw data. Fox detections appear to be knocked down quite quickly after baiting, whilst cat detection increases before decreasing. This is likely due to baiting being more effective for foxes, whilst it takes longer to get on top of the cats, using primarily trapping and shooting. From the raw counts, it looked like there may be some evidence of a trap response when cameras are first set, and also potentially seasonal effects. I'm looking at these covariates in the full data set. And now the spatial count density estimates. Firstly, the northern grid, with baiting to the right of the dotted line. Again, foxes are in the closed circles and cats in the open. Fox density has markedly dropped post-baiting. Cat density looks to have increased slightly, then decreased. Hopefully the full data set will provide a clearer picture. Next, density estimates from the southern unbaited grid and the northern baited grid in spring 2018. The fox population has been knocked down and cat density is similar despite differences in the fox density. So this is an incomplete data set and we have a lot of work still to do. We commenced catching foxes and cats for satellite tracking last year. This was disrupted due to coronavirus and hopefully we'll be able to commence in spring. The telemetry data will allow us to verify our sigma priors and provide a subset of marked individuals and their locations to better inform our observation model. I would like to thank my collaborators, funding bodies, field assistants, and fellow Quaco Lab members for their assistance in this work. Please get in contact if you have any questions or suggestions. Thank you.